In this part, we're going to review some of the basic structures that are found in eukaryotic cells. Like prokaryotic cells, eukaryotic cells are going to have some structures that are found in all of them, and some eukaryotic structures are going to be found in certain ones. And again, these differences can be used to help us to facilitate the process of identifying an unknown organism. Again, let's start off with an overview cartoon of what our typical eukaryotic cell will look like. Over here we see a micrograph, electron micrograph, which shows some of the internal structures. It's actually, I think, a lot easier to see things in a eukaryotic cell. That's probably a factor of its much larger size. Things are easier to see. But the cartoonist, the artist for the textbook, has drawn a little cartoon with labels for some of the things inside. And we'll spend our time talking about a number of these things and what role they play in the cells as we move inward from the center. Let's start off with a structure that we know already from our talk in prokaryotic cells, and that's the capsule. We recall that capsules helped in the process of attachment and help the cell to be protected from things out in the environment. And we recall that the capsule, the glycocalyx of bacteria, was made of sugars. And these are made out of sugars as well, but biochemically they're very unique, very different from those in bacteria, even though they're made of these same building blocks of sugars. We can see a picture here which shows the presence of a capsule surrounding this yeast cell, Cryptococcus neoformans. The capsule is this hollow area, this halo surrounding the cells. Cryptococcus neoformans is a serious pathogen that causes a form of infection of the central nervous system called meningitis, and it's the presence of these capsules that greatly enables this microorganism to be able to cause these infections. Some eukaryotic cells have cell walls, but this is again another optional structure found in some, but not all. We recall that the prokaryotic cell wall was made of a compound called peptidoglycan. We spent a lot of time speaking about the chemical nature of peptidoglycan. Eukaryotic cell walls are not made out of peptidoglycan at all. They're made out of carbohydrates. Here we have a fungal cell. This is the cell wall down here, and it's made up of a polymer of glucose. Just glucose repeated over and over and over again. It's this repeating layer of glucose that surrounds the cell, and it does the same thing that the prokaryotic cell wall, that it serves to protect the cell from things like osmotic lysis. But it's important to recall that it's chemically going to be very unique from the cell walls of bacterial cells. When we get into our chapter about antibiotics, we're going to learn about one antibiotic that you probably have already heard of. It's called penicillin, and what penicillin does is it affects the ability of the cell wall to be formed, but it only does it with cell walls made out of peptidoglycan, which means it's only going to affect bacteria. This yeast cell right here causes disease in humans. It has a cell wall, but penicillin is not going to have any effect on this cell wall because the cell wall is not made out of peptidoglycan. What kind of cells in eukaryotic cells have cell walls? Well, it's not all of them. Animal cells do not have cell walls, but fungi, plants, and algae all have cell walls that are chemically rather similar from one another, made out of these polymers of glucose. Now, the next part is not a structure, but really kind of a process, and it's this process of endocytosis that you may remember from Biology 1. And endocytosis is the active process. It requires energy and how eukaryotic cells get materials into the cell. Eukaryotic cells are surrounded by a cellular membrane, a plasma membrane that's essentially identical to the one that we already saw with our prokaryotic cells. And eukaryotic cells, just like prokaryotic cells, do passive transport, and they do active transport. using cell proteins, cell membrane proteins that enable molecules to get in and out of the cell. Endocytosis is a little bit special. It's going to be used for very large materials. We can see, for instance, this microorganism. This is a rather large microorganism, and this even larger eukaryotic cell is first attaching and then engulfing this second microorganism, bringing the second microorganism into the interior of the first one, where this poor little guy is going to get digested. Endocytosis is used by all eukaryotic cells to bring in large materials into the cell. We're going to see an incredibly specialized form of endocytosis called phagocytosis, which is the way that our immune system fights off invading microorganisms, engulfs them, brings them into the cells, and destroys them through a manner that's extremely reminiscent of this. But we'll use this term phagocytosis to speak of specifically how our immune cells destroy invading pathogens. Alright, so another optional structure that's found on some eukaryotic cells, but not others, are flagella, that we recall from our section on Bio 1. Again, it's optional. We spent a little bit of time looking at the makeup of prokaryotic flagella. We learned that the main proteins that were important for us were two of them. One was called flagellin, which was the protein that went to make up the flagella itself, and the second protein was a protein called ATPase, which used the energy of ATP and turned it into the energy of movement. Eukaryotic flagella are not made out of flagellin at all. They're made up of this protein called tubulin, but they also have ATP activity. We can see this sort of illustrated a little bit more closely in here. These long tubules, these bundles of tubules right here, are the bundles of tubulin, and ATP, what it's going to do is it's going to cause these tubules to slide back and forth against one another. And what that enables the flagella to do, as these tubules, these microtubules made of tubulin, move back and forth against one another, it enables the flagella to slide back and forth, and the flagella, instead of moving in a rotational motion, moves in a back and forth motion, a whip-like motion that propels the organism forward through its environment. Now, tubulin is found in the flagella of eukaryotic cells. It's found one other place in the cell, and it's not on the exterior of the cell. It's found on the interior. This very pretty looking picture right here is a number of eukaryotic cells. I'll outline one of them right here so we can see it. That's the outline of this cell. Here's a structure we're going to get to in just a minute. But these green tendrils that are extending all through the cell like this are microtubules made out of tubulin. So tubulin serves more than one role in the eukaryotic cell. It forms the tubules that go to make up the flagella that helps a eukaryotic cell to move if it possesses them. But in all eukaryotic cells, it forms these tubules inside the cell, which make up the bulk of a structure called the cytoskeleton. We recall that eukaryotic cells are very large in comparison to prokaryotic cells. Being large, they need something to hold their shape. Humans have a skeleton inside, an endoskeleton that helps hold our bodies into the shape. A eukaryotic cell has a cytoskeleton that gives that cell its shape. They also serve, these tubules inside the cell also serve another really important purpose. When we get into the next couple of slides, we'll see that this isn't just a static bag of goo, that things are moving in and around this cell from one place to another place. And these microtubules made of tubulin are critical for this process as well. If we come in with a magic drug that causes tubulin to stop working, everything inside the cell comes to a standstill as all transport in the cell is blocked. Alright, here's the big organelle in eukaryotic cells, the nucleus. We remember the nucleus. This is the storehouse of DNA. It's found in all eukaryotic cells, and in fact the word nucleus gives eukaryote its name. That term karyote means nucleus. The word eukaryote means true nucleus. Found in all eukaryotic cells, the DNA is found in chromosomes that are encased inside this membrane. The process of mitosis allows these chromosomes to be able to divide in two, and the process of cellular division produces daughter cells. But all of the information in the cell stored in here inside the nucleus. Well, although the process of DNA replication, all the DNA is stored inside the nucleus, most of the chemistry of the cell occurs outside of the nucleus. One critical function that we're going to be talking about and reviewing in our chapter in genetics is the process of protein translation, where the information in messenger RNAs is converted to proteins. That, of course, occurs with ribosomes, and these ribosomes will frequently associate with a membrane organelle in the eukaryotic cell called the endoplasmic reticulum. We can see a ribosome right here. A messenger RNA would associate with this ribosome, and a new protein would be made here in the interior of this endoplasmic reticulum. Protein synthesis is also going to occur outside of this endoplasmic reticulum, out in the cytoplasm, but the proteins that are formed within the endoplasmic reticulum here in the internal part are actually going to be very specific. They're going to be serving very specific roles within the cells. We're going to see some of the things that proteins that get made with the endoplasmic reticulum, what they can do. This organelle is one of the places that new proteins made in the endoplasmic reticulum can go to. It's the Golgi complex. The Golgi complex is a place where proteins that have been made in the eukaryotic cell can end up where they are temporarily stored and then sorted to go to other locations. Proteins move from the ER and then travel to the Golgi complex. And then from the Golgi complex, they can end up to a variety of destinations throughout the eukaryotic cell. Perhaps some of them can go to the nucleus, some of them may go to the plasma membrane, some of them may go to other organelles. One of them we're going to be talking about in just a minute, which is the lysosome, which is, as we'll see, plays a critical role with the process of endocytosis that we just saw in the previous slide. What I like to think about the Golgi complex is kind of like the post office of the eukaryotic cell. We're probably familiar with this idea that the post office acts as a sorting location for mail. What is it that enables mail to get to its final destination? Well, it's the address or the zip code that's on that letter. And what happens is that the mail ends up at the post office, and that zip code is used to dictate whether or not the mail ends up at the nucleus, or whether the mail ultimately ends up at the plasma membrane, or whether the mail ultimately ends up at the lysosome. This cartoon kind of gives a big picture of the movement of molecules through a typical eukaryotic cell. We recall that the endoplasmic reticulum is going to be playing a role in the process of protein synthesis. New proteins are made here, and then they move into the Golgi complex. And from the Golgi complex go to final destinations throughout the cell. One place that these new proteins can go to is the lysosome, which is filled with all sorts of enzymes that play an important role in the digestion of things that come into the cell. We can see this bacterium being brought into the cell through the process of endocytosis, and we can see here it's given the specialized name phagocytosis. The bacterium is brought into the cell because it's very, very large. This phagosome gets mixed up with the lysosome, and these digestive enzymes go in and kill the bacteria. digest the bacteria into small component parts. Some of the parts may be used for nutrition for this eukaryotic cell, and some of the trash gets dumped out through the opposite of endocytosis, exocytosis. And there's a number of these so-called late endomembrane compartments. They're destinations for proteins that are made by the Endoplasmic Reticulum that gets sorted to by the Golgi. The most important of these for Bio 230 are going to be the lysosomes, which, as we said, are filled with lots of different enzymes that break down large molecules that have been brought into the eukaryotic cell through the process of endocytosis. All right, another organelle that's found in eukaryotic cells is this one right here, the mitochondria. It's not optional. It's found in essentially all eukaryotic cells. It's not a organelle that's made part of the endomembrane system, the ER Golgi lysosome set of organelles that we saw in the previous couple of slides. We can see in a typical mitochondria right down here, you can see it has a relatively complex internal structure, though the names of these structures are not going to be critically important for us to know or understand about. All mitochondria in eukaryotic cells facilitate this reaction. We can see the metabolism of this molecule, glucose, plus oxygen, to form carbon dioxide and water, and this is the process of cellular respiration. Which, when it goes to completion, releases a huge amount of energy in the form of ATP that is used for all sorts of functions within the cell. Now, mitochondria then, because they carry out this process of cellularization so well, are known colloquially as the powerhouse of the cell. Now, scientists began studying mitochondria and their internal structure once powerful enough microscopes were developed, and we can see on here that mitochondria contain ribosomes. When people also further analyzed mitochondria on a very close level, they also found that they also contain DNA. They have a small DNA genome inside. And mitochondria are observed under the microscope to divide by binary fission in a manner that's very much like the division of bacterial cells. And so it actually appears that mitochondria very closely resemble and have many of the behaviors of prokaryotic cells. This will become important for us in just another few minutes. Another structure that's found in eukaryotic cells that superficially resembles the mitochondria, it's a structure found in photosynthetic organisms, and carries out this reaction where carbon dioxide and water are metabolized to form glucose plus oxygen. And note that this requires an input of energy from the light of the sun. Much like mitochondria, chloroplasts also contain DNA. They have ribosomes and produce some of their own proteins and divide by the process of binary fission in a manner very reminiscent of prokaryotic cells. The glucose that's made here by the action of the chloroplast is then available for the organism that has chloroplasts for its own process of cellular respiration. Plants, photosynthetic, they have chloroplasts. They also have these guys, mitochondria, as well, that carry out cellular respiration for it. And excess glucose that's produced by photosynthetic organisms is used by all other organisms in the biosphere. Now, the presence of chloroplasts and mitochondria with their own DNA and their own ribosomes is used as one of the key pieces of evidence for this theory of evolution that explains the origin of eukaryotic cells. What's believed to have occurred during evolutionary history is that at an early point in time there were only prokaryotic cells that did not have any internal membrane-bound organelles at all. And there was an event that occurred at some point during this evolutionary process where two different cells that were able to carry out specialized metabolic reactions began to associate with one another in a symbiotic relationship. We can see this sort of drawn here where we have one cell perhaps engulfs a second cell by the process of endocytosis until we get a cell that has all sorts of internalized organisms that are acting in a symbiotic relationship. The evidence for this endosymbiotic theory for the origin of eukaryotic cells comes about from mitochondria and chloroplasts, which is, we said in the previous two slides, mitochondria and chloroplasts, they have DNA genomes and they have ribosomes. And the other kicker with this is that the ribosomes that we find in mitochondria and chloroplasts, they don't resemble the ribosomes that we find in the rest of the eukaryotic cell, the large ribosomes of eukaryotic cells. They very closely resemble the smaller ribosomes that we find in prokaryotic cells today. And in fact, after we go and we sequence the DNA genomes in mitochondria and chloroplasts, we can identify living prokaryotic cells that genetically very closely resemble the DNA that we find in mitochondria and chloroplasts. And so it's these pieces of evidence that we use to support this idea that eukaryotic cells, the cells that we find in human beings, have arisen during the course of evolutionary development by the association of prokaryotic cells, leading to a symbiosis that's absolutely dependent upon one another. And this table just sort of summarizes the various kinds of structures that we find in eukaryotic cells. We can see these structures here present in eukaryotic cells and the structures that we find in various kinds of prokaryotic cells. The ones that we want to focus on here in Bio 230 are these, the ones that we find in the bacteria, the glycocalyx, the ability to move with flagella, the presence of fimbriae or pili, the presence of a cell wall and what makes up the cell wall, membranes, cytoplasm, ribosomes, endospores, and the presence in a prokaryote of a single circular chromosome. Eukaryotes are going to have all of the different free organelles in here, the endoplasmic reticulum, the Golgi, lysosomes, mitochondria, and chloroplasts, but lacking many of the other things that we talked about in prokaryotic cells.